All right, let's talk about the renal tubules here. So our nephron, which again is the functional unit of the kidney, contains several sections. You first have the glomerulus, which are the blood vessels, and here are the capillaries, and they are a special type of capillary called a fenestrated capillary. So that's the glomerulus. Then you have the Bowman's capsule or renal capsule right here, and together that is the renal corpuscle, okay? That leads to the proximal convoluted tubule out through here. You know what? Let's change our color here because that might help. The fluid, the plasma that comes out of the glomerulus associated with this efferent arteriole here, it literally is blood plasma, but at this point, by the time it gets into the proximal convoluted tubule, we call that filtrate because it's fluid that's been filtered from the blood, right? And so it travels through the proximal convoluted tubule, down here through the descending loop of Henle. And so you can see there are lots of little twists and turns here. Down here where it turns around eventually, so the descending is going down, ascending loop of Henle has this thicker portion, which we'll talk about in a little bit, heads back up into the distal convoluted tubule and down through the collecting duct, where the collecting duct will, excuse me, enter one of the renal calyces and eventually dump the concentrated urine. So this is urine out. Oops, urine out. And I think it's kind of funny because any place where you see urine associated with the nephron, it's yellow. So anyways, just something to think about. Let me get rid of my green lines here. So what does the tissue look like? What do the cells look like? In the proximal convoluted tubule, you'll see that you have basically cuboidal cells that have some cilia. Not a lot of cilia, but occasionally you'll see some cilia there. By the time we get down here to the descending loop, at the thin portion of the descending loop, has basically a squamous epithelium. So you've gone from these cuboidal cells in the thick part down to a real thin layer. And what do thin layers allow to happen? Escape. Remember that? That's something that we will talk about in a little bit in more detail. Let me see if I can get my eraser to work here. No. Oh, yeah, there it is. Now, going up through the ascending loop, the conformation or the organization of cellular tissue changes. Now we have small cuboidal cells. These cells have tight junctions. Stuff does not escape from this region. We'll talk about that more later. As we go up through the distal convoluted tubule, this looks a whole lot like the proximal convoluted tubule without the presence of some microvilli. They're not cilia, oops, they're microvilli. And then eventually out to the collecting tubule, excuse me, the collecting duct, which are more kind of elongated cells. They're not quite as columnar, but they're very, very tightly joined to one another, and fluid does not escape out of the collecting duct. So the first segment has some microvilli here. The loop of Henle, the extension going down, has the descending limb, the loop down on the bottom, the ascending limb, then the distal convoluted tubule, and the collecting duct. Or tubule, call it tubule as well. So what does the kidney do? We have to understand what the kidney does so that we can look inside of the kidney and see where it does the jobs that are important. So what does the kidney do? It excretes toxins. Okay, toxins can be medications, drugs sometimes, and nitrogenous waste. Nitrogenous waste typically comes from nitrogen. Nitrogen, where does that come from? Proteins. Okay, so the proteins that we digest and break down into amino acids get converted to other amino acids in our liver, and when that happens, sometimes nitrogen groups are chopped off because nitrogens are a part of the amino group. Remember what an amino acid looks like? Carbon, then there's a hydrogen, and then a carboxyl. Oops, and then an NH3. All right, and then down here, there could be, basically there's a functional group down here that specializes the amino acid. When the amino acid becomes a part of a protein or it gets converted to something else, that gets chopped off, and that it becomes nitrogenous waste. Pretty effective, right? Pretty efficient. Kidneys regulate lots of chemicals in the blood. You can worry about that later when you're in Bio-18. Kidneys obviously maintain water balance, which is a big deal. If we drink enough water, if we are adequately hydrated, we excrete water. But if we need water, if we're dehydrated, we will downregulate nutrition or urination to help maintain water in our system. And because we have a maintenance of water balance, that also helps regulate blood pressure. Because remember, where is all of the urine coming from? What is the filtrate? Blood plasma. Blood plasma turns into urine, basically. And how do you get blood plasma? You drink water. So you drink in water, it's absorbed through the digestive tract into the bloodstream where it becomes a part of the plasma. It can get into tissues where it becomes interstitial fluid. It can get into the cellular space. It can become intracellular water. Water eventually then drains into the lymphatic system, maybe back into capillary beds, and eventually it drains out through the kidneys. So water is really, drinking water is a big deal, and it has lots of different names depending on where that water lives in your body. So how is urine actually formed? So there's a three-step process that takes place, and that happens in different parts of the nephron. And again, that's why we call the nephron the functional unit. So filtration, the first step, happens at Bowman's capsule, okay, at the glomerular capsule. And so that's where, as I mentioned before, water can, or fluid, plasma, can leak out of the fenestrated capillaries of the glomerulus based on blood pressure. So if we have normal blood pressure, right, standard regular blood pressure, that blood pressure allows fluid to escape through the glomerulus, through those fenestrated or slits in the capillaries, and that allows water and other dissolved compounds, some compounds, not all, to filter out into the filter out of the glomerulus into Bowman's capsule and down into, eventually, right after that step, the proximal convoluted tubule. If somebody has regular blood pressure, that happens at the rate of about 125 milliliters per minute. So that's a lot of fluid going through a single glomerulus every minute. After filtration occurs, certain compounds are reabsorbed from the body. So you have, here's Bowman's capsule, my version of Bowman's capsule, right? Here's the proximal convoluted tubule. Let's make the glomerulus here. Here's the afferent, okay? So fluid, which I am going to make green, well, they make it yellow for urine. So you have the filtrate coming out. It comes into the proximal convoluted tubule, and here water becomes absorbed, reabsorbed, and we'll talk about where that water goes in just a minute. I'll go back to a picture in a minute. Other nutrients that might escape out can be reabsorbed here. Certain ions can be reabsorbed, but basically you have the reabsorption of water here via osmosis from the proximal convoluted tubule. At that point, we also have something else called secretion happening. Well, what is secretion? Oh, let me go back to the red pen. So if blood is coming in this direction, circulating around in the glomerulus and coming back out, now we have this blood vessel that's traveling adjacent to, and I'll try to make it look like a blood vessel, adjacent to the proximal convoluted tubule. Eventually this will dip down and follow the nephron. So the nephron will, let's say, take a turn here and go down so that it can go down into the loop of Henle. I kind of straightened out my proximal convoluted tubule. But this is called a peritubular capillary. Okay, it used to be called the vasa recta. At this point, the peritubular capillaries can start secreting stuff into the urine that'll pass through the proximal convoluted tubule. Certain drugs and medication can be secreted into the proximal convoluted tubule, and ammonia can be secreted. So basically you have some stuff that escapes out through filtration and some stuff that gets literally secreted and pumped into the proximal convoluted tubule from the peritubular capillaries. Now I'm going to go back a couple slides here. Don't go blind. Here are the peritubular capillaries, the red capillaries, all right? Like I said, before we used to call them the vasa recta, but now most books call them peritubular capillaries because they're capillaries that will secrete into, look at this, look at the proximal convoluted tubule. Look at all this space, all these places that secretion can happen. pretty efficient, right? And this is how we get rid of things like, oh, ibuprofen, all right? This is one of the places that acetaminophen gets secreted out through the peritubular capillaries into the proximal convoluted tubule. Okay, here's really the meat of what's happening. We have our three processes: filtration, secretion, and reabsorption. So here's Bowman's capsule, and they're showing fluid or blood cycling up in this direction, okay? Cycling from the bottom of the slide up. Here's the glomerulus in Bowman's capsule. The fluid escapes out through the fenestrated capillaries, and that is the filtration step right here. The filtrate, the fluid, okay, because it's been filtered already, so now we're going to call it filtrate, that escapes into the proximal convoluted tubule where water gets reabsorbed, okay? What you're going to see are a lot of places where water is reabsorbed from the proximal convoluted tubule. The descending loop of Henle, you'll see some water reabsorption happening and glucose reabsorption happening here. Okay, the kidney, if some glucose gets out into the filtrate, it helps our body to maintain glucose in our bloodstream, right? Because, well, glycolysis, right? So here's glucose. You know what? I am going to use blue because it's reabsorption. Okay, so glucose is reabsorbed, and sodium is reabsorbed, and water is reabsorbed here. Now, as we descend down through the descending loop of Henle, reabsorption doesn't happen. All right, other things are going on there. It's a lot more complicated, which you will see in Bio-18 at Merced College. But now as we head back up through the distal convoluted tubule, we have potassium and water again being reabsorbed back into the peritubular capillaries adjacent to the nephron. Secretion happens. All right, so we have secretion happening here between the peritubular capillaries and the distal convoluted tubule. And the collecting duct. So we have nitrogenous wastes, that's an amino group, right? And protons of all weird things are being secreted into the the distal convoluted tubule and the collecting duct. What does this do? What do protons do? That's acid, remember? So this is what partly makes urine somewhat acidic. So we have our processes: filtration, that happens only at Bowman's capsule, absorption, and secretion. So how is urine volume controlled during all of these processes? What does our body do to keep urine volume relatively stable depending on what we take in and what we secrete? Well, remember our pituitary? The pituitary makes antidiuretic hormone, and it is exactly what it is, anti-diuretic, right? So a diuretic would be allowing lots of water to be released to make urine, all right? Anti-diuretic means that you're not peeing that much. So I just think of diuretic, peeing, anti-diuretic, no peeing. This is also, anti-diuretic hormone is also called vasopressin. So vasopressin, not vasopressor, vasopressin decreases urination. Urinalysis is a process that we do actually at Merced College in our Bio-18 class, where we look at the composition of somebody's urine. Why would we want to do this? Well, we would want to know what is the body, in addition to the normal things that are being filtered out and secreted out, we want to know if other things are escaping into the urine. So sometimes in filtration, it doesn't normally happen, but sometimes protein will escape into the urine. Our body tries to hold on to protein for obvious reasons, right? If protein is found in urine, that could mean a couple things. Number one, in women, oftentimes when women become pregnant, there's protein in the urine. Number two, high blood pressure. High blood pressure results in higher pressure inside the glomerulus, higher glomerular pressure, which means higher filtration pressure, which means stuff gets squeezed out. Larger things escape into the filtrate. So other things that we look at in urinalysis is the presence or hopefully absence of like fungi or bacteria, which it would be consistent with the bacterial or yeast infection in an individual. And sometimes we look for little crystals, which would be the formation of different types of crystals in the kidneys, which also can be the result of infection or disease. So we will start the next part of this lecture looking at the ureters.